Merhaba Deniz Hocam, hoş geldiniz. Berfin Hanım, hoş geldiniz.
Merhabalar hocam.
Merhabalar.
Merhabalar. Bir 5 dakikamız var. Ben açılışı yaparım, sonra devam edelim.
Tamam, bekliyoruz hocam.
Hocam şimdi mikrofonunuzu kapatabilir misiniz acaba? Cızırtı yapıyordu.
Serdar Bey, hoş geldiniz Intergen'den. Evet biliyoruz, ikinci sunumu siz yapacaksınız. Deniz Hocam, mikrofonunuz kapalı.
Hocam merhaba, nasılsınız?
Teşekkürler, iyiyim. Siz nasılsınız? Pınar Hoca da burada. Onu da
Merhaba.
herhalde Berfin burayı alacak.
Aldım
Hoş
hocam.
geldiniz hocam.
Bir dakika, kameram kapalıydı. Şimdi açayım ben. Merhabalar, hoş bulduk. Nasılsınız?
Merhaba Pınar.
Teşekkür ederiz.
Merhaba Serdar
Hoş
abi.
geldiniz hocam.
Merhaba Bahar hocam, merhabalar. Deniz
Hoş
Hanım.
geldiniz Serdar hocam, Pınar hocam.
Merhaba.
Var mı? Merhabalar.
Bir de Işıl Hoca var galiba.
Evet, onu bekliyoruz.
Ondan sonra da başlayalım. Bu arada
Henüz 3 dakikamız var
katılımcılar
hocam.
arasında benim Türk Bebeği birlikte yaptığım Emine Hoca bulunmakta. Ona merhaba diyorum. Biz yıllar önce beraber Türk Bebek sertifikasını almıştık. Gelecekte benimle birlikte çalışacak olan hocamız da burada. Emine Tural Hoca. Pamukkale'den gelecek. Gene Pamukkale'den Serkan Hoca bulunuyor. Bizim kongrenin düzenleyiciden bize buna çok emeğe geçen Kenan Bey burada. Online süreçte Pınar Hocam siz de yaşamışsınızdır. Artık iyice Zoom'da içli dışlı olduk.
Evet, evet gerçekten çok böyle hani dersler artık hani ekiplerle, asistanlarımızla toplantılar bu şekilde olmaya başladı. Gerçek görüşme gibi olmasa da hani yolculuk mesafesi o zamanı atlayıp hemen online görüşebilme şansımız, saat sınırımızın olmaması da belki bir avantaj oluyor.
Ve
Mesela
aynı zamanda dezavantaj oluyor. Sağ olsun. Herkes her şeyi istiyor aynı anda.
çok doğru
Bir
söylüyorsunuz.
Fransız arkadaşımız var. O şey diyor, Zoom pandemisi bu nedenle de kafamızı işten kaldıramıyoruz artık diye. Kesinlikle doğru söylüyor.
Ben Pınar'cığım, Serdar Bey, hoş geldiniz diyorum.
Evet hocam.
Var hocam merhabalar. Sizi görmek ne güzel.
Merhabalar, Sayın Moderatörüm, Ünal Bey, Deniz Hocam.
Hoş geldiniz Bahar hocam.
Kıymetlilerimizle beraber olmak bizi çok mutlu etti, çok onur duyduk katılımınızdan.
Gülen
Buyurun
yüzünüzü
Ünal Hocam.
görmek çok güzel bir şey Bahar hocam. Gülen yüzünüzü görmek çok güzel.
Sağ olun, sağ olun.
Gerçekten çok teşekkürler.
Ben kısa bir konuşmayla başlayayım.
Evet.
Değerli katılımcılar, konuşmalarını sunacak sayın hocalarım, kongremizin genetik başlıklı 3. oturumuna hoş geldiniz. Biz bu oturumu ben Profesör Doktor Ünal Uslu, değerli arkadaşlarım Doçent Doktor Deniz Atasatar ve Doktor Öğretim Üyesi Nuray Bostancıoğlu ile birlikte yönlendirmeye çalışacağız. Oturumumuzda birbirinden değerli 3 bilim insanını konuk edeceğiz. Onların sunumları aracılığıyla biz de bilgi dağarcığımızı genişletmeye çalışacağız. Oturumumuzun açılış çalışmasını yapmak üzere değerli hocam Marmara Üniversitesi Tıp Fakültesi Genetik Anabilim Dalı'ndan Profesör Doktor Pınar Atay'ı ben sözü bırakıyorum. Teşekkür ediyorum.
Teşekkür ederim. Ben bütün katılımcılara ve gerçekten organize eden kurula, Bahar Hocama, yine moderatörlerime. Ben sunumumu bugün size hani çok bilinmeyen bir konu değil ama çok gerçekten sıcak bir konuyla ve başlayacağım. Umarım sorularımız olursa birlikte yine paylaşırız. Benim anlatacağım konu aslında gen editleme ya da gen tedavisinde durum nedir, şu an geldiğimiz nokta nedir ve bu noktada ekson atlama dediğimiz tedavi yöntemini ne kadar kullanabiliyoruz? Bunlarla ilgili bir takım aslında kritik noktaları aktarmak, çok da hani onları tanıtmak, anlatmaktan ziyade şöyle bir eğer hatırlayacak olursak DNA sekansımızı, dizimizi farklı bir şekilde değiştirebilme şansımızın olduğunu anladığımız yıllara dönecek olursak çok da yeni değil. Son 5 yılda çok hızlı koşarak devam eden bu teknolojide bir takım genlerin ortadan hani kodlanan genlerin kaldırılması,
the DNA. So we can have some point mutations, we can have targeted mutagenesis, and also we have some practices in animal models. So we have molecular or gene altering in the DNA. So, but of course in human zygote, gene editing is somewhat challenging because there might be some malpractices. That is why we have some prohibitions. Lastly, in Japan, this gene editing liberation took place, but it was only for research purposes. Of course, altering a gene is possible, but we may not always reach the target because it has to be finalized with a live birth, or we can miss the target. So changing that, of course, might result in a conclusion, a result that is not desired, we can say. So in these processes, what kind of situations we may face? Well, this is nucleus-mediated gene editing. We all know that there is the double-strand break. We create a specific DSP in the genome of a cell. And in fact, this point of break should be selected very critically. And this point should activate the cell's DNA repair mechanism. However, when we do that, well, if we have the insertion in different DNA areas, we can have non-homologous end joining. So we are targeting the repair mechanism. We are trying to activate the repair mechanism in order to eliminate the defective DNA sequence. But when trying to do that, non-homologous end joining can be the result. Or another, another result is homologous recombination, also called homology-directed repair. That is, we can have the break there, and then the DNA repair mechanism can be activated. So we have two different solutions in this method. As you can see here, this is non-homologous end joining. This is classic pathway, and there's a breaking point, and different areas are joined together. Therefore, the repair mechanism takes place, and combined protein context is bound in this process. In fact, the targeted insertion takes place in the red areas. For instance, these are the targeted parts, and this is but the result is the blue targeted, blue area that is result. So this is the insertion that we have. Therefore, in the end, this may not be the area that we had targeted at the beginning, or it can be non-homologous, or they may not match. They can be different from the corresponding strand. When we look at the homology-directed repair, in this concept, in this method, some of the repair proteins are activated in this breakpoint, and in an area we have a limited resection, and in that sense there can be homology together with the corresponding strand. The repair mechanism can be activated on the same line. And different repairs can be activated. So this gene editing, nucleus-mediated gene editing, was first started with the 1990s with the meganucleases, and in mid-2000s, zinc finger nucleases were discovered. And then in the early 2010s, transcription activator like effector nucleases or talents were started to be used. So, and in 2013, very recently, the CRISPR, clustered regularly interspaced short palindromic repeat, or Cas enzyme, was a system that was discovered in the bacteria, and this can be used in the repair mechanism, which was also worth of Nobel Prize. So what can be done by using these technologies? I'm not going to talk about all the enzymes, but I will briefly talk about the pros and cons of these methods. The zinc finger nucleotides are heterodimeric protein with multimeric sequence, and they bind to certain domains, and as an advantage, they can have the most clinically advanced platform. I apologize for this background noise. Well, we have the homing endonuclease. We can have homo or dimeric protein containing site-specific DNA recognition domains. In TALEN, we have the heterodimeric protein. Again, we have different areas of resections. In CRISPs, we have a targeted area, and we can have the repair in place. So we have some more advanced techniques such as megatalen. This is the state-of-the-art technology. Well, this is zinc finger nucleus. This is just like cutting the DNA sequence with scissors. And very specifically, you can target the area that is damaged, or the area where we are going to use our enzyme. So the zinc finger nucleus or talons can be used in the targeted areas. And we, in the neighboring area of the binding sequence, we can have the targeted pattern. And the mutant sequence is in red. So there is the double-stranded break that can be formed. And this is the break in the mutant sequence. We can have homologous recombination. And the wild-type template DNA is used. The novel template, we can have the repair of the gene sequence. In fact, well, how did we come up with this gene editing method? Well, there are transcription factors, and inside the bacteria, the viral process is eliminated. And this is the immune system of bacteria. So they can recognize DNA sequences, they can bind at certain areas, and the promoter areas, promoter sequences can be bound. Therefore, the transcriptin factors, the thallus, the transcriptin activator-like effectors can target this area. And the repeated areas can be bound, and then the transcription factors are bound, and they can, let me show you this way, they can
faktörlerin etkisi altında
affect the gene under the effect of, they can regulate the gene under the effect of transcriptase factors. So we use the different repeated areas. In fact, the genetics is combined with informatics and computer technology, computer engineering, because DNA binding codes and their calculation has become a process of our routine, I can say. So these repeated areas have specific binding patterns to the gene. So we can do the single cell PCR, and then if you're going to do the reimplantation genetic definition, for instance, if it's cystic fibrosis, then gene mutation can be eliminated by means of targeting this pattern DNA. And the correct sequence is used in forming the pattern in the area. Therefore, this is an engineering practice, I can say. And once it is bound to the repeat area, then there is a targeted mutagenesis, and two different enzymes are just bound together. And once they are bound to these sequence, then there is a break. And of course, this area is repaired by means of activation. It can be a point mutation, there can be an insertion, or there can be a deletion of this area. We cannot all the time control the results, because when we have the double strand break, it is possible to have deletion. Of course, we love this treatment method. We believe that it's a miracle solution, but I need to talk about the challenges as well. So we have a modulator, we have a suppressor and activator. So this is the targeted area. I do not want to get into the details, but when we talk about gene editing, well, this in the in vivo gene editing, and a cell is taken from a patient. Of course, we cannot do them in the embryo level right now, but in the germ line, we can do that. And these types of activities are not allowed right now, but we can have somatic mutations. For instance, in the own cell of the patient, maybe the mutation can be corrected, and then it is given back to the person. For instance, in cystic fibrosis, the alleles are the same, therefore that mutation area of the patient is received, and then the stem cell of the same person is given back to the circulation of the person, back to the stem cell. Then the mutation-free gene is going to be started to be coded, and then the symptoms can be overcome by the person. We also have RNA-based genome engineering. And of course, I'm not going to get into the details, but it was first described in 1987, and bacterial immunity was first discovered, and CRISPR was one of the most famous gene editing methods. And here you can see that this is the plasmid DNA, virus plasmid DNA is transferred, and then the CRISPR RNA has the pre-CRRNA of the bacteria. Then these are taken with the Cas proteins, and then this integrated area of the virus is recognized, and then the area that is not belonging to itself is just cut off with some scissors, and the own area is patterned once again. So this CRISPR genome editing was done in animal experiments, and then maybe it can be transformed to the cells, and it can be a chance of treatment for patients. Therefore, this CRISPR and Cas system, in fact, we have a strand, we had the double strand break, but in addition to that, we have a guide RNA. So this guide RNA just shows us as a guide, it shows us the strand that is mutation-free as a guide. And then we have the trans-activating cRNA. So in the corresponding RNA, we have the binding of Cas9 molecule. As a result, a mutation-free strand sequence is activated. We can use a repressor to suppress the target gene, or we can have an activator to have the re-regulation of the normal RNA sequence. As I said before, in gene editing, we cannot use the germline cells because there might be some off-target sequences, off-target results, or there can be a manipulation of the off-target areas, and stem cells can lead to unwanted results. That is why we can have the gene editing technology on somatic cells. So when we look at the risks of genome editing, if we skip the target, then we can have genotoxic. And if we have exon skipping, we can have different mechanisms in place. There can be enzyme-related risks, and also we can have vector-related risks. That is, the toxic effects can be transferred to the live cells of the person. There can be also delivery-related problems, such as electroporation, transfection, microinjection, so on and so forth. And then we can unwillingly induce the apoptosis of the cell. And also delivery device related problems can be seen. So I wanted to go over them one by one, but I just want to summarize them. Well, we can have genotoxicity because regional mutation is targeted. And in that case, some different areas can be integrated, and we don't want some new chromosomal changes. So in the cell cycle, there can be a slowing down. So we are trying to activate the repair mechanism, but we may accidentally lead to cell apoptosis. That is the risk that we have. Exon skipping, well, it is mostly used in neurological diseases. So the damaged exon is skipped, and then we can have the protein production even if it is quite short. Well, in the terminated area mutations, we can use that. So I'm going to skip these titles. Well, let's not exceed our time. Okay, thank you very much. So in the end, I want to say that, of course, we have some promising treatments, but I think we need to be aware of the risks, such as carrier molecules, and in terms of transfer-related risks, we need to wait for some time. Thank you very much for your attention. Thank you, Pınar Hanım. you have focused on the very hot topic. Thank you very much for your contribution. In order to use time effectively, let's have the Q&A in the end. So I'd like to give the floor to Nuray. She will deliver her speech.
Good evening. My name is Nuray Bostanceri from Gaziantep
Sıradaki sunum
presentation is going to be about Gen evaluation in emergency services, yoğun intensive care unit and complications. Serdar Ceylan.
Hocamız Ankara Tıbbı bitirmiş.
He is a graduate of Ankara University Faculty of Medicine, had his post-graduation in Erciyes University. Clinical genetics is the area where he has hundreds of articles. Yes, microphone is yours, sir. Well, thank you very much for kind introduction in the first place. Can you hear me? I hope you can all hear me well.
Serdar Hocam sesiniz geliyor.
Evet, şimdi
Teşekkür
geliyor
ederim.
hocam.
Yes, now we can hear you, sir. We can hear you. Well, first of all, thank you very much for your kind invitation. This is being a very horizon opening meeting. I really take pleasure in listening to the presentations. I would really like to thank the organizing committee wholeheartedly. Of course, we talk about the science of the future, and that is our science. I'm a clinical genetics specialist. We mainly work on rare diseases. This is what I've done all my life, but I'd like to talk about a new area right now. These genetics specialists have refrained from dealing with that matter, but this subject has become very popular these days, and this is emergency services, intensive care units, and the complications in those areas. Unfortunately, these are newly on our agenda, and this has become a matter of work for me as well. Why did this become more important? As you know, all areas of medicine are working hand in hand with genetics, and in areas in an unexpected number, we are dealing with patients within that perspective. But most of our patients are in the ICU and emergency services. Why do we need genetic information? Because when it comes to rare diseases, statistics show that 10% of the society has a rare disease, and only 1/3 of those are fully diagnosed. 2/3 of the population has other multifactorial diseases, and environmental factors and genetic factors play an important role on those, like atherosclerosis and allergies. We've been aware of this background for a very long time, and this is the reason why we always want to hear about the anamnesis and want to talk about the past chronic diseases of the patients. But catching this and detecting it may not always be possible with anamnesis, because most of the patients may be asymptomatic most of the time, and they might lack diagnosis, and this is a problematic area for us. Well, we have other areas of patients as well. One of those are those with previously diagnosed genetic and rare diseases. The diagnosed ones also have many problems in the emergency, because although they have diagnosis, there might be other problems. I mean, when a doctor is confronted with a rare disease in emergency, may refrain from making an intervention and may need to consult someone who is a specialist on the subject. And rare disease, European Union's Orphan Diseases Committee is also discussing these matters these days as to what should be done when such an orphan disease patient comes to the emergency. Secondly, due to another reason, other things might happen. The patient may come to the emergency sometimes because of the rare disease or other diseases related to the rare disease, the orphan disease. Of course, there are situations when people come to the emergency with hemorrhage. As you know, after trauma, hemorrhage is the most important problem that we see in emergency. Everybody has hemorrhage after trauma, but if there is vascular abnormalities or if there are tissue fragilities in the patients, the risk is much higher and the situation might be severer. In more than 400 diseases, we see these tendencies in the patients, and some of those diseases cannot even be diagnosed of, like factor 11 deficiency, thromboxane synthase deficiency. It's not very easy to diagnose those. And there are those who are among the orphan disease class, but who are very common among the society, like von Willebrand disease. And some of them require special treatment, like hemophilia. It requires a special treatment. And some others might be quite complicated, some other diseases. I mean, in an MR image, like you see here, if there is a head trauma in the patient, you may have hesitations as to where exactly the hemorrhage is located. This is brain small vessel disease, for example. And another problem that we have, we see this very frequently, and this is the reason why I became obsessed with emergency, is acute abdomen. More than 200 diseases can call acute abdomen paroxysmal extreme pain disorder, may have a severe course. FMF is another example. Almost 30% of the FMF patients have a history of unnecessary surgery, and angioneurotic edema patients are even more difficult to diagnose, especially abdominal angioneurotic edema are harder to diagnose. Another very complicated group is porphyria patients. Porphyria patients have important and severe acute abdomen picture, and when that happens, it is almost impossible to diagnose those in emergency. I'd like to give an example of a case. I've been dealing with this patient for almost 2 years now, 32 years old, male patient. The complaint is abdominal pain and severe and acute abdominal distension after exercise. It causes short of breath. It is so much that the patient faints because the patient cannot breathe. When the patient first came to me, I made him walk a little bit, but he really could not breathe, and we had to call the ambulance. Therefore, in the ultrasound, we saw that the walls of the intestine were thickened, and as a diagnosis we said exercise stimulated angioneurotic edema. It is nice until here because we have a clinical diagnosis. I wrote a clinical report so that he would have something in hand if he again wanted to go to the emergency. And unfortunately I realized that genetic test was not giving any results in that sense. Exome sequencing is what I'm doing on this patient right now. Although he has a report and although he was in the emergency 3 times, one of which was yesterday, last night, this patient unfortunately cannot be given prophylactic treatment. Nobody wanted to deliver prophylactic treatment because there is no evidence. In some of our genetic diseases, unfortunately, we have to be faced with this picture. C1N1 rates are okay in this patient, but now the immunologic mechanism is what we're trying to understand. I talked about porphyria. Under normal conditions, the patient looks like that with a normal face, but suddenly when there is an attack, this face might change, and when that happens, the symptoms are abdominal pain, chest pain, vomiting, nausea, fever, high temperature, and purpury. And in half of the patients, we see those symptoms, but when they are combined with skin symptoms, the patients become toxic patients. And when diagnosis is not made properly, paralysia might be observed and sudden death may be seen because of the sodium level changes, and seizure might also be another problem for that patient. One of the examples that we're discussing highly these days is traffic accident and trauma, and whether it has something to do with genetics. Yes, it might be the case, because what we see is that I may have a traffic accident, another person with genetic disease may have a traffic accident, but the response to trauma is different in us. For example, COL1A1 mutation that you have may lead into osteopenia or osteoporosis. Therefore, when this person has a trauma, the fracture research period has to be longer, and you have to look at the subject with more detail. And VWF deficiency carriers in the community is 1 out of 200. And in these patients, the period of menstruation is prolonged. Therefore, brain hemorrhage is increased in the trauma. Let's say that you see a brain hemorrhage in a patient and you saw the aneurysm and you think that you're lucky to make a diagnosis. But if this aneurysm has something to do with polycystical renal disease and you do not have this genetic information, then acute renal failure might be a risk for that patient. The patient is a candidate and you may have skipped this. This is what we're talking about all the time. What you call complication is actually another name for a partial diagnosis. Coming to traffic accidents, there are other issues which are on the agenda today. In the neurological patients have more accidents, not only traffic accidents, but all sorts of those. If there is ryanidin mutation, which is quite frequent in those patients, then a surgical intervention might be problematic because malignant hyperthermia might be observed. Therefore, the frequency, the incidence is 1 out of 200. When you listen to the anamnesia, you're just going to hear that they are lazy from the family. That's it. You cannot catch any other symptom. Rhabdomyolysis is again something that we see a lot in patients in the emergency. The tissue damages, the autoimmune diseases are increased in number. The risk of thrombosis increases with this tissue problem. So they're different from the other patients. For example, if I'm working in outpatient setting, I would wonder why not all trauma patients have a brain hemorrhage and why not all patients develop renal failure. We always make a joke, and if we say that if genetic specialists start working in an outpatient center or in emergency, they would slowly start asking whether all the patients coming to the emergency are relatives to one another. So we may see such interesting pictures as well. When should we consider a genetic disease? In diseases that are already known to be genetic, FMF, cystic fibrosis, etc., are really important. We always know that. But other than this, if there is an early onset disease, for example, MI at the age of 4 or atypical prognosis, family history, failure in treatment, abnormalities, more than one concomitant diseases, or an undiagnosed patient, genetic and immunological factors should definitely be sought in those patients. We're talking about the medicine of the future. When it comes to the medicine of the future, there are different scenarios that we hear in different meetings. Suddenly, non-diagnosed diseases World Union will discuss this even better. There was a recent meeting. Let's say that COL1A1 variant is there. In minor traumas, fractures of the bone should be considered. Let's say that this is what the report says, or Bernard-Serrier syndrome in a patient, and thrombocyte transfusion is the first choice of first-line treatment. If these are written in the reports, then it would be really nice. I just experienced something like that. A patient had a traffic accident. After 1 month, still there was renal hemorrhage, but etiology was not understood. So we thought that it could be an outport carrier. Maybe a diagnosis was necessary, and we really saw an Alport syndrome carrier in front of us. In FMF patients, in long QT patients, in thrombophilia patients, we see things similar. Pharmacogenetic factors are also important these days. If you know the disease, if you know that certain drugs are being metabolized more slowly, then you can easily and roughly understand what kind of administrations should be performed. Australia, UK, and Germany have carried out a study in some of their hospitals. All patients hospitalized in neonatal department patients were examined in terms of their exome sequences, and they got an award from the European Union. The cost was reduced down to $10,000. Length of stay in the hospital was also reduced. As they could show, they have reduced mortality and morbidity by 1/3. Contribution to diagnosis is more than 50% in first application and 65% in years. This was the result that they had with their study. 2 severe COVID cases from adult intensive care unit, for example. We receive many phone calls these days on this matter. 55 years old, previously healthy male, a businessman. This patient when first had COVID, the patient was very severe and nothing was under control. So we were called, and in 6 days, exome sequencing was done, and TREX1 nonsense mutation was found. This actually is a gene of a neurodegenerative disease, but in immune system, interferon system is also affected by that. Bilkent University is a part of an international study where interferon mechanisms are found to be the primary and the most important factor in COVID disease. Again, in Istanbul, an anesthesiologist colleague of ours had hemorrhage instead of thrombosis when he was COVID. We did the test, and combined factor 5, factor 8 deficiency was seen. Heparin had to be discontinued, and at the same time, TLR2 gene was subject to mutation. Therefore, immunomodulating drugs were advantages for the treatment of that patient. So as you see, if the prognosis is sometimes bad, sometimes good, and we have to question why that is the case. The problem is that the genetic testing is very time consuming. Therefore, we have to make and store genetic test results before we will need them. Exome genome data, if we find and store those to be used in emergency cases, how would it be like? I think it would be quite helpful. Of course, a long genetic report cannot be provided to everyone. It is not healthy to say that you're going to die to someone after Alzheimer's disease. So these should be used only upon requirement. I would like to talk about 2 things, 2 anecdotes, which I've just had this week. 3 days ago, I received a patient with intracranial bleeding at week 34. There was bleeding tendency. Alloimmune thrombocytopenia was the problem, but alloimmune thrombocytopenia treatment and the other hemorrhage tendency treatments are not same during pregnancy. We don't have data. We got the samples and we're going to work on this, but we don't know what will happen to the baby in that 1-week period. Hyperechogen bowel case was another case that we saw this week. We did amniocentesis and we're going to analyze the baby as well. We see these things in ultrasound too. Recently, I saw from the studies conducted that people who come to us with complications actually emphasize the fact complication actually means a half diagnosis. Complication rates go down when you make all the advanced and detailed analysis, because genetic diagnosis is really important for everyone these days. So in summary, genetic diagnosis is going to be an indispensable part of the emergencies and ICUs soon. In neonatal ICUs, it is being used to a great extent. Exome sequencing-like structures are there and they are being put into practice, but this requires a high speed, but we're not very used to this. But I would like to thank all my team members. The people that you see there have been working nights and days. So I would like to thank them, and I would like to thank you for giving me the opportunity to speak. Thank you very much. This was a very exciting presentation. Thank you. I'd like to give the word to Ünal Bey. Maybe he would like to ask a question. Yes, Ünal Bey. We're going to take the questions at the very end in order not to exceed our time. So I would like to thank our next speaker. And at the very end, we're going to take the questions.
Hello, I'm from Adana City Hospital. I am a histologist. I would like to invite Ms. Işıl Özer from Istanbul Faculty of Medicine, Cerrahpaşa Faculty of Medicine, and she was a pediatrist, and then she had her PhD, and she is a department head of pediatric diseases in the same faculty. So I would like to invite her to make her deliver her speech. Yes, please.
Hocam sesiniz gelmiyor ya da ben
We
mi duyuyorum?
cannot hear you, Ms. Özer.
Bize de gelmiyor hocanın sesi.
Bir aksaklık var galiba.
Işıl Hocam, mikrofonunuz açık görülmesine rağmen sesiniz gelmiyor.
Well, Ms. Özer, although your microphone is on, we cannot hear you. You can share your screen now.
Işıl Hocam, son alt köşede ses şeyiniz var. Orada test edip ondan sesinizi açabilir misiniz?
Please. Your microphone.
Hocam paylaşabiliyor mu?
Işıl Hocam, sunumunuzu paylaştınız.
Tamam.
Sunumunuzu görebiliyoruz.
Yes, we can see your screen, Ms. Özer.
Yalnız sesiniz gelmiyor
Sesiniz
hocam.
gelmiyor.
But we cannot hear you.
Şu anda sesiniz gelmiyor. Siz bizi duyabiliyor musunuz?
Can you hear us, Işıl Özer?
Vahova'ya ana sayfasına çıkarak Zoom üzerinden bağlanıp Vahova'dan bağlandığımız için olabilir diyor teknik sorunlar.
Well, you need to.
Buradan düşün, join Zoom yapın isterseniz, o zaman olacaktır.
Işıl Hocam, telefonunuza bakabilirseniz ben sizi yönlendireceğim.
Değerli katılımcılar, Işıl Hoca'yı tekrar bekliyoruz. Birazdan katılacak, aramıza katılacak. Sunumunu bizle paylaşacak. Işıl Hocam katıldı aramıza.
Işıl Hoca katılana kadar şu anda mesajları okuyorum. Genetikçi hocalarımızın konuşmaları çok beğenilmiş. Hatta tıp öğrencilerinin de anlatılabilse bu sunumlar ne kadar önemli olur denilmiş. Biz Kongre Bilim Kurulu ve Yayın Kurulu olarak 2 ay kadar sitede planlıyoruz. İndirme imkanı da veriyoruz. Eğer tıp fakültesi de ders olarak sunmak isteyen hocalarımız olursa, eğer izin alındığı takdirde prezentasyon sahiplerinden tabii ki yayın paylaşılabilir. Tıp öğrencilerinden de çok katılan var kongremize. Bugün bir mesaj daha almışız. Bugün Beşiktaş ve Galatasaray derbisiyle çakışıyormuş yayınımız. İki takıma da başarılar da diliyoruz aynı zamanda.
Bu süreçte yalnız bizim de dinleyici kitlemiz çok fena değil. Hala katılımcılarımız var. Bu teknik aksaklık giderilirse daha da güzel sunumlar olacak. Hocam, yani her iki hocanın da yaptığı sunumlar gerçekten çok değerliydi. Bir hekim olarak Serdar Hoca'nın acilde yaklaşımı ayrıca çok ilginçti. Biz şu ana kadar çok komplikasyon deyip atlamıştık. Ve atlamış olduğumuzu ben Serdar Hoca'nın sunumunda fark ettim. Aslında böyle bir şey değilmiş. Biz tanıyı sonuna kadar gidemiyormuşuz. Bunu da bir kez daha öğrenmiş olduk. Chris ise benim fakültemde dinlediğim bir moleküler biyologdan ilgi duyduğum bir alandı. Çok ilginç bir şeydi. Geçenlerde sosyal medyada gezerken ben de değişik bir şey buldum. Mesela bir bakteri plazmitine gömmüşler. CRISPR'le bir genetik kodu gömmüşler ve bir atın koşmasını, galop yapmasını sürekli çoğalmasıyla gösterdiklerini dinlemiştim bir Amerikalı bilim adamının insanından. Bu da bana çok ilginç gelmişti.
Ünal Hocam, geçen gün katıldığım bir tıp fakültesi sempozyumunda öğrenciler, tıp fakültesi öğrencileri hayallerinden söz ettiler ve özellikle genetik bilmede çok ilgi duyuyorlar. Nerede sınırı olduğunu, özellikle bu embryo editing, printing, gene editing konularında Pınar Hocam da değindi, Serdar Hocam da çok hevesliler ama orada bir şeye dikkat çekildi sonuçta. Tıp öğrencilerinin bu hassasiyetine çok saygı duydum. Dediler ki hocam, bilim insanları sınırsız şeyler keşfedebilip bilimi ilerletebilirler ama hekim olunduğu zaman orada regülasyonlar işin içine girer. İnsan yararı için kullanılıp kullanılmayacağı konusunda o zaman hekimlerin de regülasyonda direktörlükte
Tamam, şimdi bakalım olacak inşallah.
denilmişti. Hoş geldiniz Işıl Hocam.
Hoş bulduk.
Hoş geldiniz
Kusura
Işıl
bakmayın,
Hocam.
biraz geciktik.
Estağfurullah, çok mutluluk verici sizlerin burada olması.
Ben çok mutluyum. Böyle bir toplantıda konuşma imkanım olduğu için. Sesim geliyor herhalde şu anda.
Hocam,
Evet
sözünüz
hocam.
mükemmel geliyor.
Evet, geliyor hocam.
Peki, o zaman konuşmayı başlatıyorum ben. Zamanda biraz geçirdik, onun için hızlı geçeceğim.
Going to be quite fast as we are already late. So first of all, I'd like to thank all the organizers. Well, I'm going to make a presentation on rare diseases and what can be done in future. And what have the advances so far showed us today. So first of all, if we define the rare diseases, the frequency, the incidence is less than 1 in per 2,000 in the society. And In terms of developments, in terms of medications, drug developments, they are quite few in number. That is why they are called orphan diseases or orphan drugs. Well, Switzerland established an information center on rare diseases, and the French Ministry of Health made the first legal regulation, and the European Union focused on these diseases between Horizon 2020, $1 billion of funding is allocated for the research of these diseases. When we look at the studies in Turkey, well, in 2017, there was a workshop that was organized in Turkey for rare diseases. And in 2020, the legal regulation was enacted. 58 diseases are included in the rare diseases, and a majority of them are hereditary metabolic diseases. So I am, this is my specialty. I'm going to focus on metabolic diseases. So in 1902, the first defined metabolic disease is, as you can see on the screen. And of course, they increased in number later on. Therefore, some categorization took place. So some subgroups emerged. You can see them on the screen. I'm not going to name them one by one, but under these subgroups, the first treatment approach was dietary treatment. So, and this is the treatment that is used for the group where we have intoxication indications. So the intoxication type metabolic diseases is the PKU diet therapy. We also have some role model diseases for the first scanning and diet, and PKU is one of them. As I said, the first treatment was started in 1952, and all the other diseases listed here just mimicked the successes of the treatment of the first group of diseases. So cofactor treatment is another application that we have. When we say cofactor, we have mostly the vitamins. So there's an enzyme that is not functioning in the body. So we have some material, some substances that are used to activate their functioning in the body. So we have the type B vitamins, and also we have the vitamin B6, pyridoxine, that is used in some of the diseases as a cofactor. Here you can see a patient. Well, when the treatment methods are found, sometimes we have coincidence, some random findings, we can say. For instance, we have cause 1 classic homocystinuria patient, at the same time primary hyperoxaluria type 1 patient.
Piridoksini verdiğimizde ikinci
And when we gave the pyridoxine, we have seen that the UCMD, Ullrich congenital muscular dystrophy, was also treated. So the muscular enzymes were corrected, and we have seen that it was treated. I tried to share this in the literature, but we have to prove that under laboratory conditions, of course. I'm sorry, could you please distance your microphone a little bit? Okay.
Soda akciğer şurubu idrar hastalığında yine aynı şekilde kofaktör etkisinden faydalanıyoruz.
CUD diseases, we use the B-type vitamins. We have two diseases here, biotinidase deficiency and MTHFR disease. In these diseases, we don't use vitamins as cofactors. However, here, particularly in the biotinidase deficiency, biotinidase enzyme activates the biotin in the body. There are two biotin molecules in the body, and then they should be separated into two, and they should be activated in the body. And biotinidase is deficient in these types of diseases because it's 1 in every 60,000, but in our country it is as frequent as 1 in every 4,000. When we look at the MTHFR, the folate is activated. And this is also seen quite frequently in our country in terms of polymorphism and in folate-related diseases. Sometimes folate is added to sugar, and it is used as a food supplement in the countries. And maybe in future practices, maybe using them, this folate and biotin, as food supplement can be a logical solution. So enzyme replacement therapies is included in the storage type diseases. Well, the enzyme is replaced synthetically. It has been in practice since 1984. So we have in the intoxication type diseases, first disease was treated. So the Gaucher type is the first disease of this ERT practices. So it is the role model disease. Gaucher had the first treatment in 1984, and then Pompe, Fabry, and in all the other diseases tried this path, and they sometimes succeeded. And we have the Niemann-Pick diseases. And Tay-Sachs disease is also here. It seems that as if it is in the beginning state, but in our country, there is a Turkish group from Izmir High Technology University. There has been some important progress. So that is why I wanted to also list this among these diseases. And also in our country, one of the intoxication diseases is PKU. In adult patients, enzyme replacement therapies are also among the options. Not hydroxylase, but iminoaminylase is replaced in these practices. I just wanted to add this because in our country as well, there's a patented research from Gazi University. I was also involved in that study. So this enzyme was used subcutaneously, and we tried to find an oral treatment of the subcutaneous enzyme. In this enzyme replacement therapies have been developed since 1984, but of course there are some challenges in practices. What happened? Well, in the end, there is a foreign material agent given to the body. There can be some allergic reactions. In some patients, there are some neutralizing antibodies developed, and also there can be no appropriate receptor for penetrating to the cerebrospinal fluid. And then, of course, these enzyme treatments are given in infusions. Therefore, there's great dependence to the hospitals. The patient should be hospitalized, and then it takes 2 to 3 hours to deliver the treatment. When it comes to substrate reduction therapy, I'm being quite fast. I'm sorry for that. I'm trying to do my best. Well, in these metabolic diseases, when we make the definition, there is a distorted or damaged gene, and there is a missing enzyme. Therefore, that enzyme cannot work, and that a product cannot be processed, and that is stored in the body, or it becomes toxic for the body. So in substrate reduction treatment, we have the first stages of the disease. So we can just prevent that at the first stages of the disease. you can see the Gaucher type 1 treatment. In type 3, in the longer period, in the late period, we also have neurological indications, and oral substrate reduction treatment could work in some of the patients. We have Fabry disease, that is another storage disease. In some of the subtypes, substrate reduction treatment works. And of course, the diseases copy the treatment methods of one another. So tyrosinemia also copied the substrate reduction treatment. You can see the child quite happy to be able to work. So tyrosinemia is an intoxication type disease, and there can be neurological attacks, exacerbations, and the patient may not be able to work. So NTBC drug works. As you can say, some of the coincidences could work in the treatment. In fact, NTBC is a rodent toxin, so a poison used for rodents, but this is used in tyrosinemia, and some successful results are received. And there was a neurological exacerbation of this patient, and then they used this treatment as a result of which they show the results. Well, we had more information about organelles in time. Well, Golgi was investigated more, and of course it goes it travels through the body until it reaches the area, and also it needs to be labeled in the end. So during that packaging and labeling phase, if the active part inside the folded the package, then the enzyme cannot work. Therefore, this mistakenly folded part should be unfolded. So you can say that is PKU that has a response to BH4. It is a natural cofactor of this. So in some of the patient groups, at the enzyme level, there is no deficiency, and the when BH4 is given, then it can work. So this is one of the great examples of chaperone treatment. So as these studies advanced in Golgi, then as we said, they are labeling on the material. So monosaccharides just label that those structures, and whenever they do not work, whenever there is a diseased Golgi, then congenital glycosylation defects are seen. And we can see these diseases more in the form of a disease family. So we have seen that they can also benefit from chaperone treatment. Well, I already talked about Tay-Sachs, but it can also benefit from other types of treatments. There is a 3-molecule disease that it used in malaria treatment, but it can also work in Tay-Sachs disease, which was found. So the treatment can work in these diseases. Well, we have to finish because the next session will start.
En son gen nakilleri. Ben organizasyon şemasında Sağlık Bakanlığı,
the organization chart of the Ministry of Finance, General Directorate of Healthcare Services, and you can see autism is under the rare groups, but tissue, organ transplant, and dialysis services department is different, and maybe for treatment purposes they could be combined. That is my view. And also I'd like to talk about the pre-implantation genetics screening test? Well, we say that there is a regulation on rare diseases in 2020, and particularly for SMA, ALS, there is the hereditary metabolic diseases, and they should be defined. And well, COVID-19 gave us important messages. When the peak experienced in Samsun, we had this control by the police. So the enzyme treatment patients could not even come to the hospital. So instead of ERT, oral treatment at home or gene therapy or organ transplants can be used in future. And for our own patient group, why are they rare? Well, either they are low in incidence or these patients are infertile and they cannot reproduce. But in terms of our patient groups, they have siblings. Therefore, they don't have reproductive problems. So, and also they get married and they can have children. Therefore, we need to, although there is the pre-implantation genetics, they still have children. So we need to have healthy genes so as not to increase this number of abnormalities. Well, in 2019, we had a symposium on metabolic diseases. So, and when I saw your pioneers in future medicine, I have been very glad that many different disciplines come.
Konuda çözümlere daha kolay ulaşılır diye düşünüyorum. Teşekkür ederim. Kusura bakmayın, başlangıçta biraz geciktik. Konuşma uzamış oldu. Teşekkürler.
Hocamıza bu güzel sunum için çok teşekkür ediyoruz. Diğer oturuma sarktığımız için soruları şu anda alamayacağız. Herkese teşekkürlerimiz.
Diğer oturumumuzdaki tüm sunumlar video şeklinde. O yüzden soru cevaplara şimdilik devam edebiliriz. Çok uzatmamak kaydıyla.
Tamam, o zaman sorusu olan varsa alabiliriz.
Ben soru değil ama Işıl Hocam'ın bu güzel sunumundan çok etkilendim. Özellikle hem genç genetikçilerin hem genç embriyologların birlikte çalışma imkanı yaratılması ve Sağlık Bakanlığı'nın bu çalışma ortamlarını, yani entegre eğitim programını desteklemesi açısından, çünkü çok yakın iki klinik birbirleriyle hem teknik anlamda hem bilimsel anlamda hem de sosyal anlamda çok iç içe olması gereken dersler de var. Hem eğitimimizi biz genetik programları dahil ederek biraz embriyologların da bir sample isterken, genetik bir sample yollarken gelen sonuçları yorumlayabileceği eğitimde olmasını çok arzuluyoruz. Biz bütün genetik sunumcu arkadaşlara özellikle teşekkür ederken de eğitim programımızda onlarla birlikte hareket edip birlikte iletişimi güçlendirmek adına hem dernekler bazında hem akademik bazda işbirliğini öneriyoruz ve birlikte yayınlayacağımız deklarasyonlarımızın Sağlık Bakanlığı'nın da yönlendirici olacağını düşünüyorum.
Teşekkür ederiz Bahar Hocam bu güzel konuşma için. Sorusu olan mı? Şuradan soru cevaba bakıyorum ama henüz bir soru göremiyorum yazılan.
Az önce Serkan Hocam'ın bir sorusu vardı. Pınar Hoca zamanında sanırım sorulmuştu. Özellikle azospermide sanırım delesyonların çözüm olup olamayacağını genetik testlerin sormuştu. Eğer hocalarımızdan şu anda session'da olan Serdar Hocam, Pınar Hocam ya da Işıl Hocam varsa bu konuda neler düşündüğünü anlatabilirse, aydınlatabilirse çok mutlu oluruz.
Teşekkür ederim ben Serkan Hoca'ya. Öncelikle şöyle azospermide gerçekten hani mutasyonlardan çok biz bir kromozomal parça kayıplarını AZF bölgelerinde delesyonlar şeklinde daha büyük genetik bölgelerin kaybedilmesi ile karşılaşıyoruz. O yüzden hani şimdi gen editleme veya tedavileri daha çok tek gen hastalıklarında kullanabilirliğimiz şu an hali hazırda çalışmalarda daha yaygın olarak ele alınıyor. O yüzden belki hani mutasyonla inaktive olmuş bir durumu varsa o çiftin o mutasyona özgü bir şey yapılabilir. Tek nükleotit mutasyonuna genin fonksiyonunu toparlayabilmek amaçlı ama büyük parça delesyonu veya dengeli belki translokasyon taşıyorlar, belki füzyon bölgesiyle ilgili bir sıkıntı olabilir. Translokasyona bağlı azospermiyi yaşayan sadece AZF bölgeleri azospermik faktör bölgeleriyle ilgili problem olmayabilir. O yüzden de büyük parçalarla ilgili olan problemlerde aslında pek gen editleme sürecimizi kullanamıyoruz. Çünkü biz daha çok daha hedefli, daha hedefe spesifik nokta mutasyonları veya küçük delesyonlar, küçük insersiyonlar gibi bölgelerle çalışabiliyoruz. Benim tecrübelerim dahilinde. Ben diğer hocalarımın da hani eğer bu konuda bir fikirleri varsa katkılarını bekliyorum.
Bizim de bilgimiz aynı yönde. Yani Pınar Hocam'ın dediği gibi yani büyük delesyonlarla ilgili şu anda bence hani bir yeniden bir yola girilmesi gerekecek. Çok fazla gördüğümüz hastalık var. Herhalde bunlar yeni baştan bir tedavi, herhalde bir Nobel ödülü daha alacak bir konu diye düşünüyorum.
Teşekkür ederiz.
Benim kişisel bir merakım var, izniniz olursa sizlere sormak isterim. Tıp devamlı ilerliyor. Tabii genetik birimi de o şekilde. Ben sizin sunumlarınızdan anladım. Sanki lütfen af buyurun eğer yanlış bir şey söylersem. Girişimsel genetik diye bir yere doğru mu gidiyor genetik? Hani radyolojideki girişimsel radyoloji gibi bir yere mi gidiyor? Başka bir ana bilim dalına mı evrilmek üzere? Klinikteki uygulamalarınızın sizin regülasyonlarla yeterli olduğunu düşünüyor musunuz? Bu konuda fikriniz var mı?
Ben açıkçası tabii hukukla ilgili daha çok Serdar Hoca daha bilgilidir. Öncelikle sözü ona ben bırakayım isterseniz ama çok kısaca hani manipülasyonlar için tabii hukuksal gelişmelerin bu genetik alanda ve yine embriyo biliminde, yine histoloji, embriyoloji biliminde olduğu kadar hızlı gelişemediği diyelim, daha hızlı evrilemediği hukuk kurallarının kesin. O yüzden de sanırım yine manipülasyonlar gündeme geldiğinde farklı şeylerle karşılaşacağız diye düşünüyorum. Serdar Hoca belki çok daha değil mi bu konuda idarecilik falan da yaptığı için ona da danışmak daha doğru olacak.
Estağfurullah. Şimdi şöyle aslında bu müdahale alanlarımız bir anda değişti. Son zamanlarda bu gen tedavileriyle beraber hem temel bilimle uğraşan genetikçiler hem de klinik genetikle uğraşanlar biraz daha kliniklerle daha içli dışlı çalışıyorlar. Ben hani biraz yaşım ortaya çıkacak ama ben 30 yıldır genetikle uğraşıyorum. Belki daha fazla. İlk başta sadece tanı koymaya çalışıyorduk. Şimdi tanı ve tedavi süreçlerini yönlendirmeye çalışıyoruz. Aslında olay şu tarafa doğru gidiyor. Subbranşlaşmış genetikçiler ortaya çıkacak ve multidisipliner klinikler ortaya çıkacak. Yani artık biz hani bir kliniğin başına şu metabolizma kliniği, nefroloji kliniği yazmak yerine o grup hastalıklarla uğraşan multidisipliner takımları, özellikle çok organı tutan hastalıkları tedavi eden ekipler oluşturmaya çalışacağız gibi görünüyor. Şimdi Avrupa Birliği'nde Avrupa Tıp Uzmanları Birliği çatısı altında Nadir Hastalıklar Multidisipliner Komitesi oluşturuldu. Ben bir süredir onun başkan yardımcısıyım. Oradaki bütün tartışma multidisipliner klinikleri oluşturmakla ilgili. Çünkü bazen bunlarda şunu göreceğiz. Yani şu anda ben yakın zamanda TÇK ile de bu konuda bir bazı toplantılar yaptım. kişiye özgü, sadece o kişi için bir ilaç üreteceğimiz zamana doğru gidiyoruz. Dolayısıyla ruhsatlandırma sürecini bile yeni baştan tartışmamız gerekecek. İşte Işıl hocalarla biz yıllardır çalışırız. Yani onun bir metabolizma hastalığı için genel bir tedavi yoksa o zaman ona kişiye özel bir tedaviyi üretmek ya da bir süre sonra işte embriyolara müdahalenin etik kuralları konulduğunda, güvenlik sınırları iyi oluşturulduğunda o embriyoya özgü bir tedavi algoritmasını oluşturacak hale gelecek sistem. Burada tabii ne tek başına genetikçi ne sahada çalışan klinikçi tek başına bunu organize edemeyecek. Çok komplike. Bizim içinde bile branşımız, yani bizim merkezimizde şu anda 2 tane bilgisayar mühendisi çalışıyor. Yani biyoinformatikçiler çalışıyor. Artık böyle hani elektronik mühendisi transfer etmeye çalışıyorum. Yani gerçekten iş çok multidisiplinerin ötesinde bir yere doğru gitmeye başladı. Ama bu tanımlar hani çok şeyler değişecek. 5 sene içinde çok değişik şeyleri konuşacağımızı düşünüyorum ben.
Bu da kongremizin amacına ulaştığını gösteriyor Serdar Hocam. Özellikle yarın Zafer Gören hocamızın anlatacağı yeni ilaç metotlarıyla beraber genetikle, çünkü gene gun'lar belki bu farmakolojiyle birlikte belki kullanılacak. Biraz enfeksiyon hastalıklarla güncel COVID section'ıyla birlikte yapıyorlar gerçi sunumlarını, ama çok yararlanacağımızı zannediyorum eğer yarınki oturumlara da katılırsanız. Çok seviniriz. Çok teşekkür ediyorum kendi adıma katılımlarınızdan dolayı.
Çok teşekkür ederiz. Çok güzel sunumlardı. Katılımcının değerli katkılarına ayrıca teşekkür ediyoruz.
Teşekkür ederiz hocam, hocalarım. Gerçekten güzel sunumlardı. Soru yoksa oturumu kapatabiliriz. Ya da katkısı olan var mı?
Ben de teşekkür etmek istiyorum bütün hocalarıma. Çok sağ olun.
Aynı şekilde teşekkürler.
Teşekkür
Güzel bir
ederim.
toplantı oldu.
Sağ
Çok
olun.
faydalandım. Sağ olun.
Sağ olun.
Oturumu kapatıyorum. İyi akşamlar.
İyi akşamlar.